y'all, welcome back. This is lecture 2. It actually covers one tiny piece of chapter 2 and then the beginning of chapter 3. So stick to the readings that I have given you through the Canvas site and through Pearson, because we're doing parts of the chapters. You're actually not responsible for the almost all of chapter 2, and you're not responsible for things like cellular respiration and glycolysis. So absolutely, if you want to read them, go ahead and do it, but it's not part of this class. For this lecture, I'm going to cover the cell membrane. We're going to talk about how things get in and out of a cell. Remember, that was something that living things did. They regulated what came in and out of them. We're going to put some cells in solution in pictures and see what happens to them depending on what they're surrounded by. We're going to look at the structure of the nucleus and other of the components, things that are inside the cell, that in eukaryotes provide protected compartments to keep things separated from each other. We'll look at the endoplasmic reticulum, the Golgi apparatus, lysosomes, and a couple other structures inside the cell. We're going to look really one slide briefly about how cells move, and then we'll come back and talk about that later with reproduction and some other topics that we cover. I want to look at the differences between cilia and flagella, a little bit about spindle fibers and centrioles, and then right at the beginning, what I'm going to go to right now is a little bit about what enzymes are and how enzymes work, because they show up in almost every lecture as we go through this class. Before we start, are you ready? I won't do this with every single lecture, but in the beginning I will. Do you have your notes out? Do you have a way to take notes? Do you have a way to ask questions? You can email me during the lecture and I will answer you as soon as I am able, usually within immediately, but if it's the middle of the night, I'll answer you in the morning. Don't worry about sending me a bunch of random emails. I'm happy to talk to you. I always think it's weird online that I don't get to see you that much. Set up your space for learning. Make sure it's cleared off, that you have space to think and to understand what you're looking at, and you have a little bit of time to do this without massive distractions happening. The video is going to stop at several points. I think there are 8 questions in this one. Obviously, I haven't recorded it yet, but I think there's 8 questions in this one. It'll stop and ask you questions. These do count for points. Overall, the lecture questions for each lecture are worth about 3 points total for the class, but they build up over time. They are, again, really similar to what I will ask you on the exam. Are you keeping up with the class? You have your schedule here of things you should be doing. I think if you're starting chapter 2, 3 here, you should be doing the readings. It should be somewhere around the beginning of June or the end of May right now for you. You should read the one chapter in chapter 2 that I have for you about proteins and enzymes. I'm really focusing on enzymes, but I have no way to isolate that out, so just the enzyme part. Then where I highlighted readings for you for structure and functions of cells, you have a discussion assignment that is due at the end of May for you to introduce yourself, but you also have some follow-up replies that are due the first week of June. Make sure you are on target with that. Again, reach out to me if there are any problems. Let's get started. Oh, okay, we're going too fast. We're getting started. I want to talk about a little bit of what's happening in chapter 2. The way I assigned it in the readings, I had to assign the whole chapter. I'm only interested in about the last 4 paragraphs for this class. And that is that enzymes are proteins that act as biological catalysts. A catalyst is some kind of substance that speeds up a reaction. In your body, every single time you go through one part of a metabolic pathway, you go through one reaction. Whether you are building something up or breaking something down, you need a catalyst or some kind of protein normally to help that reaction happen within the environment of your body within your lifetime. These reactions are things that would already happen, but maybe not happen at a speed that is facilitating life. Basically, I have 2 words that go with this to describe breaking things down and building things up. When we talk about molecules being broken up, we talk about pathways or reactions that are catabolic. This is a cat in this movie, and the cat is knocking a glass off the edge of the counter. As if you've ever had a cat or been around a cat or known a cat, they like to knock things off of things. They're functionally catabolic. They like to break things down. Enzymes help molecules facilitate the reactions of the breaking down of molecules. But they also facilitate reactions where we build molecules together. Building molecules together is called anabolic. You may have heard of anabolic steroids. People take them to build muscle. Anabolic means to build. If I want to take two molecules and cause them to form a larger molecule by joining together, I need an anabolic pathway. And enzymes help with both of these. Enzymes are proteins, mostly. I have one enzyme that is not a protein. I'll introduce it as we go through the rest of the lecture. These enzymes that are mostly proteins, they cause reactions to happen faster in your body. They are not causing reactions that are biologically impossible to happen. They are causing reactions that would normally happen if we left the molecules alone for 100 million years. They cause them to happen in seconds or milliseconds within your body. The way they work is they fundamentally lower the amount of energy that you need to enter into that reaction to make it happen. Every time you want to break things up, catabolize, or build things together, anabolize a reaction, you need to add a little bit of a startup energy to get it going. Enzymes lower that amount of energy that you need to activate a reaction. We say they lower the activation energy, or if we want to really shorthand it, I can write it like this. This is the activation energy. There's a weird delay in my clicking forward. Bear with me here. What that means, and this is a chart I took from a totally different textbook, just to show you the reaction here, and I've edited it a bunch. The reaction here is showing this is an increase in energy on this axis, and time is proceeding this way. If I start out with these things, these are the things that I want to react in my body. They are my reactants. And I want them to be turned into products, or the final result that I want. I have to give a little bit of, this represents a little bit of rise in energy. This is energy I need to put into this reaction. When it happens normally, that's purple line here that I'm tracing. That purple line requires this much, a whole bunch of energy in order to get me to the point where the reaction will then just proceed on its own to the products. When I add an enzyme, the amount of energy that I need is lowered. This is the energy of activation. I don't need to add this much energy. I only need to add this much, which is much lower, to have the same result and get to the end of my products. Enzymes facilitate, they make reactions happen faster. Enzymes speed up reactions by lowering the barrier of how much energy I need, the energy of activation. There's a couple other rules for enzymes I want to point out as we go along. These will help you as we move through the course. They make reactions happen faster. They do not get used up. So when I add an enzyme in my body to facilitate breaking something up, maybe in my stomach when I'm breaking down proteins, those enzymes just keep working as long as they have the same structure to themselves. If I want to stop them, I either have to break the enzyme or I have to block it so it doesn't work anymore. They lower the reaction, the energy, this is energy of activation, by actually physically bringing together the things I want to react, or by pulling apart the things I want to break apart. They are physically changing the structure of the thing they are reacting with each other. Every time you hear a chemical named and it ends in ase, lipase, sucrase, peptase, that ase ending at the end of the word is a hint to you that you are looking at an enzyme. It's not going to hold 100% of the time, but 99.99999% of the time, ase is a clue that you're looking at an enzyme in the metabolic pathway we're talking about. They can't make an impossible reaction happen, but they do make reactions happen faster. Enzymes work kind of like a lock and key mechanism. So I want to quick ask you a question, and so I'll pause so I can do that, and then I'll finish up the idea of an enzyme here. Enzymes work like a lock and key mechanism. They have what's called an active site. They have a physical shape in the structure of the molecule where the reactants actually squish in and fit with them. If the reactants don't fit, the enzyme doesn't do anything. If the enzyme's shape is different than it was supposed to be, and its shape of that site where the reaction takes place, or its active site is different, the enzyme doesn't work anymore. The things that fit into the active site, the reactants, during an enzymatic reaction, we call them the substrates. The substrate is the thing that the enzyme's active site acts on. So the active site here is where all the action happens. And the stuff that goes into it, the reactants, are called the substrate. I did not name these. I would not have named them like this. I think it's a little confusing, but I'll talk about active sites and substrates because that's how the vocabulary works. I want to move, which is a little abrupt, I want to move from chapter 2 and our just highlights of enzymes, because we need that stuff, into chapter 3 where we talk about what is a cell. One of the ways we defined life at the very beginning of the class, so in the last lecture, was that cells are the smallest unit of life. We have this idea of cell theory. Real fast, I want to ask you another question. What's a theory? Because cell theory is the idea that all life is made out of cells. All living things are comprised of cells. A single cell is actually the smallest thing that can be considered alive. If you're less than a cell, you're a molecule, or you're an atom, you're not considered alive. And all cells come from previous cells. Cell theory is the basic idea of how we fundamentally understand life to exist. There are two broad categories of cells. We talked about cells that were in the bacteria and the archaea in the first lecture. Those were prokaryotic cells. And we talked about cells like you. You are a eukaryotic cell. Eu meant true. Karyotic meant nucleus. You're a true nucleus cell. Kind of FYI, because it's not really part of our class very much, but pro means before, or the first. Karyote meant nucleus. Bacteria and archaea are prokaryotes. They are cells that evolved before the nucleus evolved. But whether you're a prokaryote or a eukaryote, you have the four following things. Every cell has a plasma membrane. That's an outside that protects it from the outside world, keeps stuff inside the cell that's supposed to stay in, keep outside stuff outside the cell, and then it's selectively permeable, meaning that some things can move through. It's made out of phospholipids. I'm going to highlight that in the next couple slides. So give me a minute and I'll get to the phospholipid bilayer. But its basic structure is fat. And those fat cells then, those fat structures protect the outside layer of the cell. Inside the cell is cytoplasm. You'll also hear it called cytosol in other classes. In this class, I'm going to call it cytoplasm. Cyto means cell. And plasm is the plasma membrane. It's the cell, the stuff inside the cell's plasma membrane. It's a semi-fluid substance. It has a lot of sugars and enzymes in it that make that all the cell's metabolism work. All cells have DNA. If you are alive, you use DNA as your information structure. DNA is packaged in chromosomes or a single chromosome, depending on what you are, and that's made out of nucleotides that carry information from one generation to the next. And all cells have ribosomes. Ribosomes are little structures made out of protein and RNA, which is like DNA but slightly structurally different. And their job, their function, is to build proteins. They catalyze the reaction that causes the building blocks of proteins, amino acids, to become functional proteins. Ribosomes are our only example of an enzyme that is not made out of completely proteins. Ribosomes are also enzymes. They're called technically ribozymes because they're special. Prokaryotic cells, things like the bacteria and the archaea, evolved before the nucleus evolved. They tend to be really tiny. You don't need to memorize this, but this is really, really tiny. These are 10 nanometers or less. You can't see them with your naked eye. They do not have any containers inside of them. And that means that they have to stay really, really small. They do not have a nucleus. Instead, they have DNA unbound in their cytoplasm, in their just fluid on the inside. That DNA is in a little loop-de-loop usually. If I draw a bacteria here, sometimes their DNA is found sort of in a little single circular loop-de-loop. Yep. This is called a nucleoid. This is something you only find in prokaryotes. They do have ribosomes because all living things have ribosomes. They float around free in the cell solution or the cytoplasm, the cytosol. They are used to catalyze building proteins. They follow the instructions of the DNA to build the proteins that that body or that cell needs to build. They divide and reproduce by a process called binary fission, which is very simple compared to what we do. These cells basically copy their DNA and pop in half. Binary means 2, fission means separate. They pop themselves into 2. And part of the reason bacteria can become so abundant so quickly is that they're fairly simple and they reproduce by this really fast splitting in 2 mechanism. Not all eukaryotic cells are even remotely the same. So I want to introduce the basics of eukaryotic cells, but then as we go through the class, I'm going to add things and take away things depending on what kind of eukaryotic cells we are talking about. Our cells are alive, and so each cell in your body is alive, but they actually function together to make you function. If I take away a muscle cell from you or I take away a nerve cell, it cannot live on its own. It is part of a multicellular organism, you, and each one of those cells then kind of specializes in doing one particular thing to make you, the whole organism, work. Your muscle cells are full of mitochondria. They have a lot of the powerhouse of the cell, meaning they make a lot of energy. And so they're really good at contracting and moving you or pumping your heart or moving food through your digestive system. Your nerve cells are long with extended parts of the cells on them. They're really good for sending messages. In fact, they don't contract like your mitochondria. They don't have as many as your muscle cells. They don't have as many mitochondria as your muscle cells because they do something totally different. In this far right one here, this is a lining of a tube inside of your kidney. We'll look at that very soon, actually. These are specializing in moving things from inside to outside of the kidney. Their shape is very different. Their structure on a cellular level is very different because they have a different kind of job. Generally, though, eukaryotic cells, and if I look at things that are plants, animals, fungus, and even the protist, we have some things that all eukaryotic cells have in common. They're typically larger than prokaryotic cells. They have their DNA in a nucleus. I'm going to zoom in on the nucleus in a couple slides and talk about what its real function is, but it's holding on to often many chromosomes, many long strands of DNA that are found in sort of a linear structure, lines. There are additional organelles besides the nucleus, things like mitochondria in all eukaryotes, chloroplasts that do photosynthesis in plants, as well as some of the structures we're going to talk about in the endomembrane system, or the inside, endo, the membrane, system of containers that allow the cell in a eukaryote to have a lot of different functions happening at the same time. We've got ribosomes because everybody's got ribosomes, and we can have those ribosomes like floating around in the cytoplasm, or they can be bound into the endomembrane system. And I'll show you some highlights of that. But if they're bound into the endomembrane system, that often means that they're doing stuff for the rest of the body, not just that particular cell. And when eukaryotes go through cell division, we do a process called mitosis, which is a lot longer and more complicated than binary fission. It takes us longer to make copies of our cells because they're bigger and more complicated. Cells have to stay small, though. If you're a cell, you can only get to be so big and still be functional. Have you ever been walking down the street in the city and a giant cell rolls down the seat and it's like sucking people up and people are screaming? No? Yeah, no, that never happens. And part of that is because cells just don't work when they're really big. They have that plasma membrane on the outside. It's semi-permeable, which means it's a selective barrier. It lets some things in and some things not in. Things like oxygen and nutrients and waste are allowed to move through that membrane in various ways. But once a cell has a lot of surface area to the amount of inside stuff it's got, it becomes very difficult to maintain the movement of things like oxygen and nutrients and waste that are needed to be maintained for that cell to keep itself alive, to do all of its metabolism. If you want to get bigger, there's some options. If you want to get big like a eukaryotic cell, you can add containers and specialized places where reactions take place isolated from the rest of the cell. Or you can become multicellular, like us, where we got really, really big because we added a lot of different kinds of cells doing different jobs. We have some cells that are specialized in one role or another. We can add little outgrowths to our cell. These are microvilli, like the lining of your intestines actually has all these little finger extensions on it. Micro means small, and villi actually means little hairs or growths off of them. This allows this cell to have all of this extra surface area on top. But at some point, a cell can only maintain, I don't know, maybe about something you might be able to see with your naked eye, like a frog egg. But most of them are still must be too small for us to see, just to maintain their metabolism. So the nucleus contains the cell's DNA. It is a container inside of the cell where the DNA is held and protected from the surrounding cytoplasm of the cell. This is partially an advantage to avoid viruses getting in. If a virus gets in your cell, it really wants to get at your DNA. If it doesn't get at your DNA and you keep it protected, that's your code that makes you're kind of winning in that situation. If something gets in and mutates your DNA or damages it, you lose your code that makes you you. It's very important to us as eukaryotes to protect our DNA inside of our nucleus. If we look at it with a microscope, I see a nucleus here. It's surrounded by actually a phospholipid bilayer, a little fatty layer of phospholipids. And there's this little dark blob on the inside. When I stain it with an inky stain, it stains much darker. This is because this surrounding area out here is full of DNA, and the darker blob here is a different molecule, so it picks up the stain differently. This molecule stains darker. It's RNA. I'll do this here. as opposed to DNA. RNA helps DNA read the code. DNA by itself is just the code for you. It's like a textbook that you never open. RNA allows you to read that code and utilize it to build your body's function. RNA is also used to build ribosomes. Ribosomes are found in all cells. DNA is found in all cells. Ribosome is found in all cells, and it takes the code that is read from the DNA and catalyzes its conversion into a protein. The DNA is read inside the nucleus. It's converted and translated or transcribed into a little tiny message called messenger RNA, a short segment of RNA that's small enough that it can leave the nucleus itself. It ekes through little doorways in the nucleus, and it goes out into the cell's cytoplasm and slides itself into a ribosome. The ribosome is also made out of RNA. It reads the message in the messenger RNA, and the ribosome then catalyzes the building of a protein by putting together the building blocks of proteins, amino acids, following DNA's rules. For this class, that is as much as I want you to understand about that process. If you have questions more detailed about that, I absolutely can answer them. This is a big part of our modern understanding of how genetics actually works. This is the expression part of like how you actually express your DNA. The ribosome helps you do that. All cells have ribosomes. In eukaryotes, some ribosomes are free and some ribosomes are bound. Free ribosomes means they're not tied to any organelle. They float around in the cytoplasm, and they generally are used to make proteins that are going to stay inside the cell. Other ribosomes are bound. Bound means that they're tied into some of the other organelles. They're normally tied into an organelle called the rough endoplasmic reticulum, which is the next slide. The rough endoplasmic reticulum, with the help of the ribosomes, helps prepare the proteins. The ribosome makes the protein, it dumps it into the endoplasmic reticulum, and then inside the endoplasmic reticulum, it's prepared for shipment outside of the cell. The endoplasmic reticulum is made up of two parts. The rough and the smooth. Rough just means that it looks bumpy when I look at it with a microscope. I'll hint at how cleverly these are named too. When these were first seen by people looking with microscopes, they could see the nucleus, which they thought looked like a little container inside of the cell. So they thought that must be where the brain of the cell is, and it is. That's where the information is. And then they could see these wiggly back and forth things. Some of them looked bumpy and some of them looked smooth. The rough endoplasmic reticulum's name is truly the bumpy inside the plasma membrane, and then reticulum means sort of back and forth. So this organelle is called the bumpy inside the cell back and forth thing. The bumpy inside the cell back and forth thing, the rough endoplasmic reticulum, is involved in protein synthesis. The ribosomes make the proteins, and then the endoplasmic reticulum modifies those proteins. It folds them into their final form so that they have the right shape to do their final function. The other part of the endoplasmic reticulum, the one that didn't look bumpy, they called the smooth inside the cell back and forth thingy, the smooth endoplasmic reticulum. It's involved in other molecules that the cell is working with. It synthesizes, means it builds lipids, those are fats, and hormones. Hormones are also built on a cholesterol or fatty lipidy structure. It stores calcium ions. Ththese are Ca2 positive. These are ions, charged atoms that we're going to use for things like signaling in the body as well as muscle contractions. And it actually takes some poisons and detoxifies them. It's very abundant in your liver, where the liver is doing things like maybe detoxifying any drugs or alcohol you take. And by drugs, it could be antibiotics, it could be Tylenol, it could be anything that you take inside your body, fats, sugars, and it helps to break those molecules down. When the endoplasmic reticulum is completed its job of modifying that protein is packaged into a vesicle. A vesicle is a small container from the ER, the rough ER. The product or the protein that was just made travels in a vesicle to the Golgi apparatus. You may have learned this as the Golgi bodies. It's the same thing. It just depends on who wrote the textbook. The Golgi apparatus or the Golgi bodies takes that folded, finished, mostly protein that the rough ER helped finalize, and it packages it and it prepares it for shipping outside of the cell. The Golgi apparatus is made up of these folded series of different containers. They're actually not connected to each other. Each time one of them, this one is receiving a piece from the endoplasmic reticulum, the vesicle travels from the endoplasmic reticulum over here to the Golgi, and then it smooshes in until the inside of what was produced in the vesicle dumps into the inside of the Golgi apparatus. The Golgi apparatus finishes the tagging and processing of it, produces another vesicle at the end, and then that vesicle is transported to the plasma membrane, where it leaves the cell. And in our bodies, it's maybe going to the blood to transport throughout the body. There's a couple other organelles that are incredibly important in the story I want to tell you about the human body. These are vesicles or somes. Vesicle means usually that it's for storing or for transport inside of the cell. Somes means that they were just named little tiny bodies. Somes. These are little tiny bodies that they saw. And bodies like containers, not bodies like human bodies. Secretory and endocytic vesicles, they store products or they secrete products that the cell is involved in. If they're secreting means it's coming out of it, they store or send out things that the cell is making. If this is a cell in your pancreas and it's making insulin, the secretory vessels store insulin, and then when your body calls for and needs insulin, the vesicles ook it out into the bloodstream, they release it, and that allows your blood levels of insulin to rise so you can maintain your blood sugar. There's also endocytic. Endo means in, cytic means cell. These are vesicles inside the cell that bring in things from the outside of the cell and store them for later. This could be molecules that the cell needs to break down. It could be a signaling molecule that the cell is going to use for later that some other cell made. Down here, the peroxisomes and lysosomes are containers that are involved in maintaining the living metabolism of the cell itself. Peroxisomes contain peroxide and other enzymes that get rid of waste material so that the cell doesn't fill up full of waste because the cell is making waste as it's doing its metabolic processes. Or lysosomes. Lyse means to break. And I've got them down here. I tried to put the vocab at the bottom here. And the lysosomes are like cell stomachs. They digest things that the cell has taken in. I pulled this picture off of Wikipedia a long time ago. This cell is eating and it's storing the food. And then it's got a lysosome here. A lysosome that is helping to digest the food. And then this cell is getting rid of waste. It's doing just about everything that's going on inside of this story. Finally, the mitochondria are incredibly important because they are the powerhouse of the cell. I was going to go with some memes here, but I decided not to. What they really do is break sugar down and convert it into cell energy. Cell energy is generally found in a form called ATP, adenosine triphosphate. If this is your thing and you're really interested in this, you can continue to read the rest of chapter 3. I'm not covering it as part of this class, but your mitochondria are incredibly important in making sure that you have enough energy to do everything from lay on the ground and not move all the way to running an ultra marathon. Your mitochondria are providing that energy in a form that your cells can use. They do that by taking in oxygen and sugar and breaking that down and converting it into something more useful to you, ATP. They do not work without oxygen. If you don't have enough oxygen, so you're suffocating, what will happen is your mitochondria can't perform the most part of their process that they do to make ATP, and your body runs out of energy. And then if you continue to not have oxygen, then you die from lack of energy. Cells can move. We'll have a couple ways inside of your body that cells are moving. Inside the cell, they have a cellular skeleton. It does not work the same way ours does. It is dynamic, meaning they can assemble it and disassemble it at will or at need. They have also movement, the ability to make themselves move in space inside of the body or to allow things to move past them. The lining of your airways and your breathing system, we haven't talked about it yet, so I don't want to give you all the words, but the lining of your esophagus and the lining of your bronchial tubes have cilia on the cells that actually move substances through that process. Things like sperm have a single long tail. If I draw a sperm here, it kind of looks like this. If I draw a cell with cilia, they'll be all over the cell. The flagella is one long tail that moves the cell in one direction. You also have structures inside your cells that help move. Centrioles are guiding the spindle fibers. That help structures move around inside of your cell and direct things like the movement of your chromosomes during mitosis so that you end up doing it correctly. Outside of the cell, you have a plasma membrane. This is a good place to take a stretch. I am trying to keep this as brief as possible, but I have these 2 lectures that go into this topic. But I want to quickly talk about the plasma membrane, and then we'll jump into osmosis and diffusion and bulk transport. The plasma membrane is the outside of the cell. It's made out of phospholipids. We can draw them like this. They kind of look like they're a head with 2 tails. They have a head that loves water. It mixes well with water, and it will turn itself towards aqueous solutions, which is most of your body. And then they have these tails. These tails are made out of a long chain of hydrogen and carbon. They are fats. They hate water. They are hydrophobic, or they are afraid of water. These tails meet in the middle. So when I look at the structure of your cell membrane, it kind of looks like tails in the middle, heads out, sort of like a peanut butter sandwich. This is the bread, two pieces of bread, and this is the peanut butter in between, the fat surrounded by some water-loving stuff. And this is enough to actually keep the structure of your cell as a little round ball. Now, obviously, you're not just made out of fat balls, or you would kind of disintegrate as it got hot out. Your membranes are a fluid mosaic. This is a mosaic. This person is working on restoring a set of tiles that when viewed from a little bit further away, they're just different colored tiles smushed together. But as you back away, you can see that there's an image in here. There's a peacock and some grapes. If we got really close, it would be hard to see because what we would focus on is each one of these individual tiles. Each one of the components of your cell membrane does one function, but together they create a mosaic of functionality that makes your cell membrane actually work. And so a little less pretty, but same idea. You have your phospholipids, lots and lots of cholesterol, which you need in order to hold your membranes together. Embed it with things like lots of proteins, doing jobs, signaling molecules and ways to recognize that this cell belongs here. All of the little molecular structures inside of your cell membrane work in a mosaic that is fluid. They can move around. They can kind of ooze one way or the other, and they create a living functional cell membrane from that. That membrane is selectively permeable, meaning some things can come out and some things can't. There are two ways that your cells control what can pass through that membrane. Your plasma membrane is selectively permeable, so it needs to control what comes in and out. It can just let physics take over. That's called passive transport. It requires no energy on the part of the cell to do passive transport. And if your cell can do something passively, it will, because it doesn't require any energy cost. There's also times when your cell needs to do things that is kind of against the way that physics would just let it happen. That's called active transport. Your cell will have to invest energy into making what it needs to move across the membrane move. I'm going to do passive transport first. There's diffusion, osmosis, and facilitated diffusion. They sound the same, but they're slightly different. And then I have 2 or 3 slides about active transport that require energy. We will use these a lot as we go through the class, because we're going to talk every time we want to move something or make something happen, it's either going to be passive or active. Passive doesn't require energy. Your go-to passive is diffusion, and you can see this every day in your life. This is the tendency of molecules to move from a place where they are highly concentrated to kind of spread out in the room. The book has a diagram of passive diffusion by putting ink in a container. You can totally do this at home with food dye. Get a cup of water, set it on a counter where it won't be bumped, and drop 2 or 3 drops of food dye in it and leave it alone. You'll notice at the very beginning, the food dye will go down in a drop and it won't go anywhere. But as time passes in the next 45 minutes or so, that color will spread throughout the entire container. And when you come back at the end of the day, you'll have a uniformly colored water without you doing anything. That's diffusion. This is not practical for an entire body to regulate things. It only works over very short distances. But things like oxygen and carbon dioxide can passively diffuse in and out of cells, and we're going to take advantage of that when we're trying to do things like breathe. We'll draw oxygen into our lungs, but then diffuse it into our bloodstream because our blood has less oxygen than the air around us has. When you reach the point where it's at balance and the molecules are evenly moving back and forth, and they don't stop moving, but they're moving at the same rate all the time, we call that dynamic equilibrium, and your jar will have a uniform color to it. Osmosis is a very specific case of diffusion. Osmosis is diffusion of water through a semi-permeable membrane. You have to say the whole thing. This is the whole definition. You can't just say diffusion of water. It's water moving from one side of a membrane to another. And the reason it does this is because it's being drawn by something that's dissolved in the water. Water will move from an area of high, or why does it say high? Ignore that. Of low solute concentration, the thing that is dissolved in the water, to an area of high solute concentration. And what that means in everyday language is that salt sucks. If you have salt or sugar or something dissolved in the water on one side of a membrane and nothing dissolved on the other, so here's some sugar is dissolved on this side, here is a membrane that is semi-permeable, and here is just straight up water. The sugar or the salt will suck the water to the other side until there is exactly the same amount of water molecules per dissolved stuff in the water on this side as there is on this side. That will cause the water to move to the side, and it'll be higher on this side, higher than on this side, which kind of looks weird. If you look at it, the water gets sucked to the saltier side, or the sugarier side. This results in osmotic pressure. Osmotic pressure is the amount of fluid that is required, of the pressure of that fluid, to oppose osmosis. We're going to use this in your blood flow. We're going to use this in how lymph builds up in your system, and how you regulate fluids packing around the outside of your body cells. I have some words to go with this that I will come back to with kidneys and the blood and lymphatic system. We have the idea of stuff that is dissolved in something else. If something is the dissolver, where things dissolve well in a substance, we call that the solvent. In biology, it's almost always water that is the solvent. So we call water is the universal, mostly solvent. The solvent in most of the cases in your body is going to be water. That's what stuff dissolves in. The solute is the stuff that is you are putting in the solvent. And this may be if you're making lemonade juice. Not today, it's freezing. But if you're making lemonade in the summer and it dissolves, you take the powder and dissolve it. The lemonade is the solute. The water is the solvent, with a T on the end. In our bodies, it's often going to be salts that are the solute, or sugar that is going to be the solute. Water moves toward the higher concentration of solute. Salt sucks. But osmosis is always about water moving. You're going to want to move the salt, but your body can't do that. It can only allow the water to move in a passive way. Oh, my joke has suddenly appeared. Tonicity is how we talk about the amount of solute in a solution. If the amount of solute in the cell and outside of the cell is the same, so here there is 9 grams of salt in a liter of solution inside and outside of the cell, that cell will be considered in an isotonic solution. It's the same tonicity, the same solute amounts inside and outside of the cell. If the cell is in a hypertonic, hyper means above or higher. If it's in a solution where there is more salt outside or more solute, what will happen is all of the water will leave the cell and the cell will shrink up and wither away, because this cell is now in a hypertonic, a much higher solution of solutes than what is inside the cell, and the water will just osmosis out. It'll suck out. If the cell is in a hypotonic solution, it means that there is more salt in the cell or more solute in the cell than there is in the surrounding liquid. The water will pour inside of the cell and it may actually explode. Boop, 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 pop. I always remember this because hippos are like really, really round and fat, and they're floating in fresh water. And it's because water is always pouring into hippos, and it's hypotonic. Not really true, but that's what the hippos look like to me. I heard they're actually all muscle. Facilitated diffusion sounds like diffusion, but there's one extra step. When things are going to move in a way that does not require energy, they're going to move from a high to a low concentration. They might not be able to get through the phospholipid bilayer, but we can still facilitate or help their movement by building a protein door. Channels and facilitated transport can move things like sodium or calcium ions. Sodium is Na, and calcium ions is Ca. These are charged particles. They cannot move through the membrane itself. But if I make a little ion channel for them, they can pass freely down from a high to a low concentration Things like sugars, like glucose, also move through channels in most of your cells, so they can kind of just flow into a cell. If the cell doesn't have any glucose, glucose will tend to enter the cell through a facilitated diffusion protein. Active transport is the opposite of passive transport. Active transport is when we require energy to move things, often against what we call the concentration gradient. It's moving from an area of low concentration to an area of high concentration. So if I am trying to fill up a cell with excess sodium, more and more and more sodium, or I'm trying to fill it up with more and more potassium or calcium, at the point where there is more calcium inside of a cell than outside of a cell, if I want to keep adding calcium in, now the cell has to use energy to do that. Cell energy is ATP. There's other kinds of energy we can use, but cell energy, ATP, is the one we're most likely going to use to power active transport. Our example is going to be the sodium-potassium pump. The sodium-potassium pump pumps sodium out. Get out. And it pumps potassium in. Okay, come on in. To the cell. And it does this in a way to maintain the cell's ability to send signals. We're going to see sodium-potassium pumps in muscle and nervous tissue cell. Any sort of cell that needs to send a signal, we're going to keep them ready to signal by using a sodium-potassium pump, which costs a lot of energy to constantly push sodium out against the concentration gradient and bring potassium in against the concentration gradient. Finally, we might want to bring in something bigger or get rid of something bigger, sort of back to the idea of how cells bring in vesicles or push out vesicles and containers. That's called bulk transport. Bulk transport requires a lot of energy and a lot of proteins working together to create movement inside of the cell. The cell skeleton might be involved. We're going to take vesicles and create them by drawing in the plasma membrane and creating a container that is made out of bent-in plasma membrane or phospholipids and some proteins. Bringing in huge amounts of things is called endo, inside. Cytosis. Endocytosis is inside the cell bringing in. There's two versions. We're going to talk about it. Phagocytosis is when the cell brings in large particles it's eating. And pinocytosis is cell drinking. This isn't osmosis. It's more like drinking water that's full of electrolytes and salt, so the cell can bring in lots of charged particles that way. Pinocytosis is more like drinking Gatorade than drinking just fresh water. The cell can also get rid of things by drawing a vesicle closer and closer and closer using the cell skeleton to the edge of the plasma membrane, and then basically smushing it into the plasma membrane until the contents of what was in the vesicle end up on the outside. That is a type of bulk transport called exo. This is outside. Cytosis is cell. We're getting rid of things that the cell had made, products, and expelling wastes. When this happens, often for you, it means you're pushing molecules into your bloodstream so that they can be transported around your body or removed if they're being filtered out. With that, that's a lot. I realize it's a lot. I have some homework for you to practice the tonicities and some of the diffusion. It's homework's more geared toward diffusion and osmosis and bulk transport than the other parts of the lecture. And I think I have one question about enzymes on the homework. Remember, we're learning this so we can apply it to the body later. All of this is words that I need to be able to use to explain things like how your muscles actually work or how your bones actually move things and keep you alive. They're actually alive and doing things for you. So take some time and make sure you are comfortable with these vocab because they all come back later. After you are happy and done, you've done your homework, you've answered all the questions to your satisfaction in this lecture, you've practiced your dynamic readings, you've read through everything, go ahead and start chapter 4, which is lecture 3 in this class. It does not look like there's a lot here. There's a lot here. We're going to learn about tissues, so we're going to put the cells together and head up a layer in our organization of bodies. We'll talk about epithelial or lining tissues, connective tissues, things like blood that hold you together, muscle tissues that make you do contractions, and nervous tissues for sending signals. And we're going to highlight some similarities and differences between them. And we're going to come back again to homeostasis, which you know from the first lecture, but that balance of making sure that your metabolism and all of your systems are within living parameters. With that, go ahead and take a break. Absolutely take a couple days off and come back, and I'll see you in the next lecture. All right. Bye-bye.